What are SNPs in arthritis research? Imagine tiny switches in your DNA that can change how your body works. These switches are called single nucleotide polymorphisms, or SNPs. They are the most common type of genetic variation among people. In arthritis research, scientists focus on these tiny differences because they can influence who gets arthritis and how severe it becomes. SNPs are like single-letter changes in the genetic code. They happen at specific spots in your DNA. Some SNPs are harmless, but others can affect how your immune system functions. Since arthritis, especially rheumatoid arthritis, is an autoimmune disease, these genetic differences can make some people more prone to developing it. Researchers have found that about 60% of rheumatoid arthritis cases are linked to genetics. That means your genes play a big role. They look at SNPs in genes that control immune responses. For example, genes like PTPN22, IL-23R, TRAF1, and CTLA4 have variations that can either raise or lower the risk of arthritis. One of the strongest genetic markers linked to rheumatoid arthritis is found in the HLA-DRB1 gene region. Certain versions, called alleles, like HLA-DRB101 and HLA-DRB104, carry what scientists call the shared epitope. These increase the chance of developing the disease. Other versions, like HLA-DRB113 and HLA-DRB115, also contribute to risk. Another important area is the TRAF1C5 gene on chromosome 9. If someone has even one copy of the risky version, their odds of developing rheumatoid arthritis go up. Having two copies makes the risk even higher. Variations in genes like CTLA4 and PDC1 also play a role. They influence how the immune system is regulated. SNPs affect arthritis risk by changing how the immune system recognizes and attacks the body's own tissues. Some variations make the immune system more likely to attack joints, leading to inflammation and damage. Others influence how much inflammatory molecules are produced, which can speed up joint destruction. Interestingly, some SNPs can actually protect against arthritis. For example, variations in the ARAC2 gene can lower the risk. This shows that genetic factors are not always about increasing risk. Some can help prevent disease. Knowing about these SNPs helps doctors predict who might develop arthritis. It also helps tailor treatments based on a person's genetic makeup. For example, some SNPs can tell us how well someone might respond to certain medications. This is called pharmacogenomics and can make treatment more effective. Genetics and environment work together. Even if someone has risky SNPs, avoiding triggers like smoking can lower their chances of developing arthritis. Conversely, people without genetic risks might still develop the disease if exposed to certain environmental factors. Scientists continue to find new SNPs linked to arthritis through large studies, called genome-wide association studies. As research advances, understanding these genetic differences will help create personalized plans for prevention and treatment. This ongoing work promises to make managing arthritis more precise and effective for each individual.